All right, so today we are going to start a new chapter, microarray data analysis, and we will specifically focus on clustering of microarray data. There are some complicated clustering problems like biclustering, which tries to find clusters of genes or clusters of samples on a subset of the samples or the genes. But we are not going to consider these complicated clustering problems. We will just see simple clustering techniques, and we'll see two algorithms: the k-means algorithm, which you probably may know if you have taken another course like machine learning or statistical data analysis. So clustering is also considered as unsupervised learning, and k-means is probably the most widely and simple clustering algorithm that is taught in these classes. And I'm also going to talk about self-organizing maps to cluster microarray data. So we will see two algorithms in the context of this chapter. And today I'm going to basically introduce you to microarray data, its nature, and what kind of public data available out there. And not only microarray data, I'm also going to show you in general transcriptome data, which shows us microarrays are things like these. So this is a microarray chip, which I analyze a lot actually. This such kind of chips. This is human genome. If you can read it here, let me get it close to you. The focus is not very bad actually, so it's not autofocus. So it says human genome U133 plus 2.0. And here, the interesting thing here is this window that you see, this square window that you see here contains around 20, I think close to 25 million pixels. So it's like you take a 25 megapixel image of this small area. There are 1 million probes. A probe occupies like a 25 pixel area here. And each probe is a small genomic sequence, usually like 25 bases, which are like signature sequences that are printed onto the chip. So there are 1 million such probe sequences that are printed here, and it has ports in the background. I think one of its ports is just sealed to maybe prevent leaks. What you do is after you create your sample that contains messenger RNAs in it, so you extract messenger RNAs and then you amplify the amount of messenger RNAs by cDNA, complementary DNAs, and then you basically inject that cDNAs at the end of this port, and you let this chip rotate around itself like this for a couple of hours, maybe 8 hours or so, that to make sure that the sample touches everywhere on this small window inside. And then you put this inside a machine to take a high resolution image of this small area, which at the end will tell you whether your sample contained messenger RNA that bound to that probe location. And then the cDNA that you construct during this amplification process contains a special molecule, like it may be biotin, which was when excited with laser, it's going to light up here. So it's like a reporter gene. It will have a certain color, a fixed color here. In this case, this is a single channel chip. There are other microarray chips available which use competitive hybridization, which means that two different samples, messenger RNAs from two different samples are dyed with different type of dye and poured together onto on a chip, and they try to compete with each other in hybridizing the probes on this chip. So This is a single chip basically used to measure the amount of messenger RNA for all the genes in a sample, all the genes that are expressed in a sample. So you the sample that you collect may be coming from wild type, normal tissue, or cancer tissue, or tissue that has been um, exposed to some hormone or some treated with some hormone or treated with some drug. So whatever cell state that you are trying to diagnose, you basically kill that cell, extract its messenger RNA at a certain, so you freeze that cell state at that moment, and such a chip gives you a way to hack to understand what messenger RNAs were there in that cell at that point. So it gives you a way to this basically the analogy in computer science is like you get a memory dump of the cell. In the memory, if you have which functions were currently active, which processes were currently running in that operating system, this such a chip is going to tell you which genes were currently active or expressed in such a state in that cell. And there are about 50,000 transcripts, 50 of 25,000 genes. All of them are on this chip. I told you that there are 1 million probes. Each gene has like they're I'm not going to go into details. They're match and mismatch probes, but each gene is represented by at 22 different positions on this chip randomly. So the value that we read takes into account noise. Um, we basically get some samples of, even for a single gene, we collect messenger RNA amount on different positions. Like 20, I told you 22, but 11 of them are match probes, 11 of them are mismatch probes. Only the most analyses use just the match probes, in which if the messenger RNA is there, the messenger RNA is likely to bind to the one of those 11 spots on this chip. And by taking average of those 11 spots, the intensity, we can get a quantitative value about how much messenger RNA was there in that cell at a particular moment. So this is really a nice dynamic analysis that shows us how genes are active in different conditions. Maybe we may use them to compare disease state with normal state, or we may have some time series kind of analysis to. By the way, this chip is one time use only. After you get the image, you just throw it away. That's how I got this actually. There were lots of thrown out chips that we could take as souvenir, and I attended a workshop, and after that I got this used chip. You cannot reuse it again, and I think it's a single chip like this is like between $200 and $400, so it's really costly. But the good thing is that, let me show you a website, NCBI Geo, in which the resulting readouts for thousands of such chips. So these are samples around. By the way, this gene expression omnibus at NCBI also contains not only microarray chips like this, they also contain next generation sequencing data. This is, as I told you last time, this is like an old technology these days. The messenger RNA amount in cells can be identified also by using next generation sequencing techniques, which provide you a higher resolution quantification of those messenger RNAs, and maybe also identify certain isoforms, different variations of message transcripts could be also identified by using those next generation sequencing techniques. And the samples that are indicated here also contains these new next generation techniques. Now, let me, for example, let's go ahead and search for, this was, the chip's name was Human Genome U133 plus 2.0. It's one of those platforms. A platform is a particular type of chip or technology. So when I search for Human Genome U133 plus 2.0, that was the name of the chip. So many results, but let's click on this result count. So here is that platform name. So when I click on that platform name, as you can see, Matrix Human Genome U133 plus 2.0 array. So this platform is entered here in NCBI Genome. And when you look at this, the description on this web page, it provides a description of this. For example, it contains probes for 47,000 different transcripts. A transcript is a particular, so it's basically messenger RNA, messenger RNA that is transcribed in the cell. All probe sets represented on the gene chip Human Genome. There's another version of this chip. They are replicated in this one. So it's from 2000s. Have some technical details about this, but here's what I want to show you is that, so this, I told you that this single chip is like $200 to like $400. There are currently 162,000 samples available. So this is a single chip. 162,000 of this particular chip has been analyzed, and the data, it's the readouts of these messenger RNAs are reported, and they're freely available. Each of them, each of these GSMs corresponds to a single chip like this. So when you click on a particular one, let's see what we're going to get. So this was left ventricle biopsy. So it was from some biopsy tissue. I don't know what this cardiomyopathy. So it's probably some disease. Familial cardiomyopathy, left ventricle. So it's part of heart. It's heart tissue. Changes in cardiac transcription profiles brought about by heart failure. So it's part of a big series. So here, this is a single sample that we're looking at. Samples are usually, when biologists conduct these analyses, they compare certain samples with other samples. They have replicates of this. A single sample from a single tissue would contain lots of variation that are due to some other things. In order to reduce the variation coming from other factors and looking only for the variation with respect to disease, we need to have a certain number of samples. The sample size, we need to have replicates data. And these samples, usually a number of them are used together in an experiment, in a biological experiment. And these experiments are, in this NCBI GEO, they're organized in a series. So here, this is the name of the experiment, GSE. It was conducted in 2004. And it contained 107 samples. So 107, and they used two different versions of chips. Probably they had in their hand two different chips, or maybe again, to reduce the variation with respect to a specific chip, they conducted again as a different technology for technical variation, to reduce technical variation. Maybe they used two different type of chips. Here, we have 107 similar chips that were used in this experiment. So it was a really expensive experiment, but if the good thing is that all this data is publicly available for researchers to use and reanalyze to find new things. So this chip, after it's analyzed, what we get here is this table. Okay, so this table, this is the actual quantitative value we're looking at. Some of these are control probes, but there are 54,000 rows here, each corresponding to some particular transcript, particular messenger RNA. When you say view full table, this is our full table. As you can see, these are, for example, let's take a look at what this is. This is a particular probe set ID. When we search for that, what it is, it says gene info. It's actually, so this is the label in data set. This is its accession number in terms of gene name, enter as gene ID. So this is the RBBP6 gene. It's actually retinoblastoma binding protein 6. So here, this entry in our, the entry that 520 as the intensity says that this RBB gene was active. So if you look at these values, they range from 0 to some large numbers like thousands, but a value of 520 tells us that this gene was present. By the way, this is this p values and p and a calls are present absent based on, you can ignore the absent calls here because either the variation between those 11 probes are too much, this is not a reliable reading, or this is a reading which doesn't, which we are not very confident. So this is the p-value that tells us we're not very confident at this reading. The p entry here tells that you may be confident at that reading. So this was the one I looked at, which tells us that gene was active in this tissue as the tissue was sampled. All right, so as you can see, this is a really rich data for a single chip. This gives us the memory dump of that, the activity level at that cell in that particular moment. So this is the data for a single chip. Usually, we conduct analysis. As you can see, there are thousands of values here for thousands of different transcripts. This single chip, the value in this single chip usually is not sufficient enough by itself without comparing with another thing, comparing with another, for example, a biopsy from a healthy tissue, or biopsy from a different tissue, for example. So what we do is, so this was the GSC. Here I'm going to show you another data now. This raw data, not the raw data, but let's take a look at the series matrix file, which is this one, this 10 megabyte file. Actually, most of the data is coming from this type of chip. And let me download this one. And I'm going to then go over the slides, but I just want to show you the nature of data that we will be looking at. So it's downloading, right? Yeah, it's finished downloading. Let me extract it here. So this is the txt file. Let me open it with. So this is a 10 megabyte text file, which contains certain metadata in it. So it was public on 2004, and there was some update in 2019. So here is a summary of the experiment. To establish changes in cardiac transcription profiles brought about by heart failure, we collected myocardial samples from patients undergoing cardiac transplantation whose failure arises from different etiologies. So there are lots of medical terms I don't know. Idiopathic dilated, so there are different causes for disease, I think. So heart failure. And from normal organ donors whose hearts cannot be used for transplants. So I was wondering how this is difficult to get biopsy from normal heart, right? I mean, if you have really a fully functioning heart, why would you go into biopsy? I mean, you don't want to get part of your heart to be taken out as to give as a normal sample here. But this is really good. It shows that if there was an organ donor, unfortunately that donor died, and his or her heart is going to be used for transplant. But if those hearts cannot be used, they can be good normal tissue samples. If nothing else, since they will not be able to use, probably there's certain time that they can be used as transplant. If they cannot be used, those tissues can be collected for later usage, and those normal tissues were obtained like this. So now we can compare two cases here: normal cardiac tissue, normal heart tissue, and heart biopsy from diseased hearts. So these are all the samples that we have here. It's not 107. These GSMs only contain the chips that use the same technology. So some details about who conducted these experiments, in which laboratory, where, and then the sample also has certain titles, maybe for identifying which patients is coming from. These are sample accession IDs. So these are some metadata we have here. And all of them are left ventricle biopsy, total RNA, again, sample description is the same. The interesting thing that I want to see here is this. So if you look at the data after this point, so I was very close. Harvard, it was conducted at Harvard University, still the metadata content, yeah, here, right here, we started saying, so this is series matrix table begin. So this is the first row of the table that gives us the column descriptions. And this is the first column ID ref, saying that this is that particular ID that corresponds to a specific transcript, a prop set which represents a transcript, which may be a transcript of a gene. And in all these different samples, we see how much transcription and the quantity that it was transcribed in all these about 100 samples. In 100 samples, in some of these it was transcribed like this, and this is another gene, how it was, the amount of transcription it had in these 100 different samples. In other words, we have ourselves, as a result of an experiment, we have ourselves some data that is showing some samples here, around 100 samples. In each of them, a particular gene's transcript, let's call it transcript. A row corresponds to a particular transcript in this series matrix that we see here. It's a transcript you see here. And what we have here are numerical values that tells us how much it was transcribed in a particular sample. Now, if these samples were organized in such a way that, for example, the first 50 of them are coming from normal, the other 50 are coming from disease hearts, we can then ask for differential express transcripts, saying that, for example, if a certain gene shows a different behavior, a certain transcript shows a different behavior in normal samples compared to the disease samples, it may be an indication of something's going on, something's different going on in the disease sample compared to the normal sample. So such kind of an analysis is called differential expression analysis. Differential expression. And there are lots of tools like EdgeR, DSEQ that does this in RNA sequencing experiments. There's also a tool, let me show you a tool, TM4MEV, this one, which is also available for use online. You may upload this data and try to, let's actually try to upload this series matrix there. Let's see whether we'll be able to do that. So get started, and you say get started. I don't want to sign up, upload a data set, tab separated. So when we're uploading tab separated TSV file, the first row is assumed to contain headers and the column gene names. So let's try to upload this series matrix file directly. Let's see whether it will be able to process this one. But with this, what you will be able to do is you'll be able to conduct analysis to see which out of these 50,000 transcripts, maybe 100 or 20 of them are really significantly differentially expressed. The differential expression can be in two ways. For example, it may be absent in the normal sample and present in the disease sample, or it may be present in the normal and absent in the disease, okay? Such a thing that depending on if you're making this analysis with respect to a disease sample, it may be upregulated in the disease sample, or it may be downregulated. So this upregulation and downregulation with respect to some disease state tells us that in the disease state it's upregulated, means that this gene, there's something that in the cell that makes this gene active. In the disease state, it's upregulated, and in normal heart tissues, for example, this gene is completely inactive, maybe, or it may be active at a small amount, but upregulation means that it's more and more active, maybe two-folds, three-folds active in that disease state. And downregulation is vice versa. It may be active originally in the normal state. In the disease state, it has repressed and not active as much as the normal tissue. So let's see, we'll be able to, so it's still processing. Let's see if it will be able to process. I think it should be, I mean, these series matrix files are really common. If it's not able to process, it's very easy to convert this notepad file into a tab separated file that it's looking for after you. All we need is copy the entire entry from here all the way to the end and paste it in Excel maybe, and then save it as a tab separated file. But in any way, this TM4, let me, yeah, actually it's now available. So let me write in the chat box the link to this tool. So when I click on it, it will hopefully tell me, yeah, the gene set, dataset, dashboard, sample sets, gene sets. So I think I should, well, yeah, dataset column heading. So it was able to process. These are my samples. If I click on my genes, wow, so it's actually doing certain analysis. Gene MAD row sets. Or let's click on this. Yeah, look at this. So it actually identified certain probes that were differentially expressed, I think, from, so this is, it conducted certain analyses automatically. But you can also, these are some analysis that you can do, statistics, clustering. So in this course, we're going to see clustering, but if you're interested in differential expression analysis, when you click on this statistic, you can conduct classical statistical tests like ANOVA, t-test. These are EDGE, R, and DSEC that I mentioned. If we click on EDGE, R, for example, when I say analyze, name is required, so we need to give it a name. Some EDGE, R test. Let me call it EDGE, R test. What is this experiment that I cannot select? I think I need to still, while processing EDGE, R analysis, I think I need to first group these genes sample sets into groups in order to perform EDGE, R. So you just need to read the manual how to conduct this analysis. But it's possible to analyze this entire data set. So this data set, as you can see, this certain color with certain color coding of these quantities that we see here, you're able to visualize it in some heat map kind of thing. We can look at the histogram or other analyses could be done with this tool. But let's go ahead and talk about more detail about these microarrays. How is this performed? How do we have these microarray quantities measured and put into these numbers? I'm going to talk about these, but this data set is really, as you can see, freely available, and it's is a result of thousands of dollars of experiment that you can analyze and maybe find other things that the researchers did not put reported in their papers. You can combine, you can conduct actually analysis that combines multiple different experiments that are available on NCBI Geo2. If you go to NCBI Geo, there are lots of Uh, Lots of experiments that are, look at the series, 5,431 different experiments performed on using this chip type, this platform type. Only this one. There are also experiments that are conducted on mm, different platforms as well. So this one expression project for oncology. If you want to find how in cancer these transcription levels change, you can take a look at this one. Probably this is a very big data set since it's part of a big project. If you look at this, there are 2,000 samples. Look at this, the cancer tissues from very different cancer types. You may analyze this data to see different expression levels between different cancer types. They should also have maybe, do they have normal non-cancer samples here? I'm not sure, but usually uh, such an analysis is done compared multiple conditions with each other, and one of these conditions has to have the control. The normal case is usually used, but yeah, NCBI Geo is a very good, valuable resource if you want to analyze these transcriptomic changes in cells that go under in different states. All right, so why do we study these messenger RNA changes? Because the messenger RNA, in turn, is there in the cell to synthesize proteins. So measuring the messenger RNA is an indirect way to measure protein levels. Because why don't we directly measure protein levels then? Because it's more difficult to, in the proteome level, the entire understanding the entire proteome and whether all the proteins in the cell, the amount that they are active is really difficult. The technology to do that is much more difficult. If you're focusing on a single protein, you can do that. There are experiments that there's a couple of proteins to localize them. They also see their amount, but doing it at the entire proteome level using mass spectrometry may be more expensive, and in certain cases it might not be feasible. So this technology of microarrays basically it promises to approximately find protein levels. I say approximately because after the messenger RNAs, there are also post-transcriptional events that may disrupt protein synthesis. It doesn't always mean that having messenger RNA in the cell may not always indicate having a certain specific type of protein at that amount, but it's really, they're correlated. The amount of messenger RNA most of the time is correlated with protein levels. Therefore, it's a good way, a good starting, although indirect, a good starting point to get which proteins are currently produced in the cell. And why do we do that? Because although our genome is fairly constant throughout different tissues, we have the same source code, same genome inside a single person's tissues. The actual running of this execution of this DNA is different from tissue to tissue because we have different functions in different tissues. The muscle tissue does not have the same function as brain and nerve cells. So therefore, it's really dynamic how this genome is executed, which parts are processed in a specific cell, in a tissue, in an organ is really dynamic. This is called this differential gene expression. Genes are expressed differently in different cells. This is what it makes different tissues different from each other. Okay, so that's the dynamicity inside a multicellular organism. In even single cellular organisms, there is differential gene expression even in E. coli because it will, based on different environmental conditions, it will require certain proteins to be made. So this dynamics is differential gene expression. And about 40% of human genes are expressed at any time in a normal cell. And here genes are expressed by transcribing DNA into a single-stranded messenger RNA. So this messenger RNA, therefore, is named the transcript. And then in the ribosome, by the ribosome, this messenger RNA is then translated into a protein. And microarrays or RNA-seq experiments, the next generation sequencing experiments, they measure the level of messenger RNA expression. There are different quantities of messenger RNA in the cell, and the more quantity we have messenger RNA, it means that the cell wants to make more of this protein. Why does the cell want to make more of this protein? It may be because certain biological processes active at that point. So by analyzing the levels of messenger RNAs in the cell and by analyzing the changes in those messenger RNA levels, we may make some conclusions about what's going on in the cell. Which biological processes are active, what is going on that is not supposed to go in a cell. Okay, so we can identify the differences between normal and disease states. So that's the, as I said, messenger RNA, and proteomics also represents the dynamic aspects of the cell. And it can be measured. This says latest technology, but as you have seen, it's available. These microarrays are available since 2004. The data set we have seen, the Geo series, was deposited on NCBI Geo in 2004. So it's been available almost for the last 20 years. And in order to measure the messenger RNA accurately, a certain protocol has to be followed. First, messenger RNA is isolated from the cell, extracted by certain enzymes, and it's amplified by using some fluorescent material. As I said, it may be biotin. I think in F-matrix gene chips, do they use biotin to do that? So here is a description of that. I think as labeling, it uses biotin, if I'm not mistaken. Yeah, biotinylated complementary RNA using RNA transcript labeling reagent. The thing is that they can be seen as some light. When they are excited by laser, they give some fluorescent colors of some fluorescent material, which means that you can measure the amount that bound there. The messenger RNA is hybridized, which means that it sticks to some target on the chip. These targets are some sequences that are printed there. Genomic sequence, some DNA that is printed there with reverse complementarity, just like the double helix of the DNA. That complementary DNA hybridizes to that target, and basically it sticks there. And there are multiple copies of cDNA at that particular probe, which allows more and more copies of the messenger's complementary DNA to bind to that spot. More DNA binds that spot means more light is going to be eliminated at that probe location, giving us an idea of, so here is an actual image. So what you see here, it looks like a noisy image. A raw image is going to look like this, but lighter colors here indicate hybridization. Darker colors indicate no hybridization at those spots. So this is, as I said, like a 25 megapixels image that is a photograph that is taken, and this is processed by, this raw image is processed into cell files, which tells us for every 25 pixels, what is the intensity for every 25 pixels. And then each 25 pixel, which we call them a probe, 11 of them form a probe set for a particular transcript. Those 11 values are averaged, the outliers, the minimum and maximum intensity values are thrown out. The remaining 9 are averaged, and most algorithms. There are basically, let me write in the chat box, two algorithms to convert these values into a single expression value, single quantity value for a transcript. The most widely used ones are RMA and Mass5, but they are not very complicated, as I said. RMA and Mass5. If you take a look at them, they normalize each array independently. So Mass5 uses data from mismatch probes, but RMA does not even use the mismatch probe. It just takes the average of those probe sets. But at the end, forget about all the detail how RMA and Mass5 works. At the end, what we have ourselves is such an expression series matrix file, which tells us this transcript has this value. It is expressed this much amount. This is the intensity at this sample. This is its intensity in the other sample. So for this is a matrix in which the number of columns is equal to the number of samples we have, and the number of rows is equal to the number of genes or number of transcripts that we are analyzing. All right, so we are going to be working with such matrix data in this part of the class. So it actually shows off the same chip that I showed you, if a matrix gene chip array. So I didn't measure it, but this window here happens to be 1.8 centimeters in each direction and 18 micrometers. A single location for a probe is very small, and there are multiple sequences that are printed in that location, which allows when messenger RNA sample is poured inside here, it allows hybridization to these spots, which in turn will will highlight those regions. So this is called single channel microarray data, because a single sample is poured in and it just gives a particular type of intensity. We just get intensity. There's also another technique called competitive hybridization, which is actually an older technique than this one, which allows comparing two different samples on the same chip by dyeing one sample with a different color than, for example, this may be the disease case, this may be the control case, and they compete the messenger RNAs from two different samples are then combined together after they are dyed with separate colors, and they are poured together into the chip, and they compete with each other in hybridizing to these locations. And if you look at this competitive hybridization image, certain parts are going to be red, which means that in the red sample, in the sample that is dyed with red, the messenger RNA is upregulated, all right? Because we see red spots here. Green spots, on the other hand, will mean it was upregulated or it was downregulated in the red sample. All right. So this is, I see some parts are red, some parts are green, some parts are totally black, which means that it was not expressed in any of these two conditions. Some parts are yellow. Yellow is like a combination of red and green. So it's maybe expressed equally in both the cells tissue types that are dyed with red and green. Okay, so this is the microarray image result, and each of these spots, they may correspond to a particular transcript again. So lots of green spots, some red spots here, some other red spots. Okay, so but the matrix-like organization of this chip has nothing to do. So this is still like two samples, normal and disease. The matrix-like organization of this, there may be different parts maybe corresponding to different transcript. Each dot here corresponds to a transcript. You should not confuse this kind of a matrix with this type of a matrix. Okay, so in this matrix, every column is a separate sample. So each of these, in other words, each of these matrix images, each of these square image, this square image that you see here corresponds to a single column in this matrix. That's a single column. So this matrix-like organization of your transcripts has nothing to do with matrix-like organization of this one. This whole matrix that you see here, row image, corresponds to a single column here on this matrix. All right, let's get back to this one. So it's about the technology. There are some papers that if you're interested in learning more about the background of microarray data, how it's processed. If you want to learn more about RMA and MES5, for example, on the course website, I have some microarrays introductory paper, and I've shared with you the username and password at the beginning of the semester. So it's CENG 465 and password is 5710465 _. So it talks about there are more example competitive hybridization images, how they are labeled, what kind of things that they measure. So you may take a look at these introductory papers. Seeing more than spots. The data in the column. So a column of the series matrix file. If you go at line 63 of this, let's go to line 63 of this file. 50, yeah, almost there. 60, yeah. If you look at line 63 of these are the column description. So each column has a label like this. The entire column is what you see here on this. The entire column is one chip. Basically, the entire column in this matrix corresponds to an image like this. All right, so this image is processed and 50,000 transcript expression values for 50,000 transcripts are obtained from this single image. Okay, this single image is turned into a single column of expression levels for all those transcripts. So those 50,000 transcripts that you see here at the first column, those are the transcript IDs. They are randomly distributed on this two-dimensional image. They have probes, 11 probes in different parts of this image. And after this raw image is processed, we get ourselves a single, the expression values at a specific state, which is this single sample. So one column is one of these images. All right, so these are our FFHX. There are different types of, you may take a look at this demonstration which shows competitive hybridization. Let's see whether this link is still active. My slides may be outdated, but let's see if it's going to work or not. Accept the risk and continue. Yeah, it still works. DNA microarray. So flash animation. Flash is no longer supported, right, in these things. So probably that's why it's not shown anymore. So, but you may see microarray methodology animations, and these days you may take a look at the YouTube video to learn more about this for the demonstration. That's basically the idea. And the experimental conditions, we may have all these columns. I told you that half of them may be normal, half of them may be diseased hard tissues, but they may be different things. They may correspond to different developmental stages, different disease states. There may be subtypes of diseases. They may correspond to different treatments, different drug treatments, different hormone treatments. So the experimental conditions really is unlimited. They're limited by the imagination or the biologists. So the experimental conditions therefore really differ a lot when they design their experiment. I'm going to just focus on, I'm not going to go into details of different microarray types. We're just going to assume we will be working on a series matrix file like this, conducted on a specific experiment with a number of samples like this and a number of transcripts. Each row corresponds to a transcript in this data. So we are going to analyze these. I'm going to stop here. I'm going to continue with this characteristics of microarray data. So I can actually show, this is a nice zoomed in version, upregulated, downregulated. If this is the experimental cDNA that we are referring against, if it's, we are analyzing this disease, which is labeled with red. If it's red here, it means that in the disease, that gene is upregulated. The messenger RNA for that gene is more in the condition that we are studying. If it is green like this, it means that in the condition that we are studying, it's not, there's no amount of red at all. So it's totally green. It means that it's downregulated. It's expressed less. If it's yellow, it's both levels. Okay, so I'm going to continue with slide number 13 on Monday. We are going to see some clustering algorithms, k-means and self-organizing map clustering algorithms on Monday that will work on a data set like this. All right, that's it from me for today. I will stop recording now.